Hello, everybody. Thank you for attending my talk today. My name is Sophia DeLuca, and I'm a PhD student in the Bursack Lab at Duke University. Today, I'll be sharing with you my research entitled Discovery of Cardiac Mitogens using a CRISPR-based high-throughput approach. To understand why we're looking to discover cardiac mitogens, it's important to understand the proliferation capacity in an adult mammal's heart. In some animals, such as zebrafish, the heart regenerates robustly after resection of up to 1/3 of the ventricle. But in adult mice, when an injury occurs, such as a heart attack, a scar forms with little to no cardiomyocyte proliferation occurring and no regeneration. However, the proliferative capacity of cardiomyocytes is inherent in mammals because if you look at studies in neonatal mice or neonatal humans, there is some regeneration and cardiomyocyte proliferation that occurs after injury at very early postnatal time points. We hypothesized that by manipulating these pathways, which are expressed in neonatal mice and turned off during development, we can induce cardiomyocyte proliferation potentially in an adult cardiomyocyte. There are several mitogenic pathways that are already known in cardiomyocytes, namely the ERBB2 pathway, the YAP pathway, and several other stimuli, including reactive oxygen species and the extracellular matrix. However, we hypothesize that there may be additional effectors either on these pathways or other pathways that can influence cardiac proliferation. So we're interested in discovering these additional mitogens. Another thing we're interested in our lab is tissue-engineered cardiac patches and their therapeutic relevance in vivo. So what you can see on the left here is a rat heart with an implanted engineered cardiac patch overlaid on the heart. However, this patch therapy has some limitations, including limited graft size. So what you can see here on the right, in green is the host myocardium, and then in green above it is the engineered human cardiac patch. So the idea behind patch therapy is that it may one day be a regenerative therapy that can aid the heart after damage. But because the graft size is so limited, we'd like to be able to proliferate this patch to increase the graft size. So that could be a potential application of our mitogens. The system I'm going to be showing you data from today is neonatal rat ventricular myocytes, or NRVMs. This is a system we can use in vitro to study cardiomyocyte proliferation. And we can use these cells in both two-dimensional culture and also 3D engineered cardiac tissues. What you can see on the left here is a graph of EDU incorporation in these cells over time in culture. And EDU is a marker of DNA synthesis, so it's a marker of cycling cells. And what you can see on the right is H3P expression, which is a marker of mitosis. Both of these cell cycle markers decrease over time in culture, such that after 3 weeks of culture, the cells are largely post-mitotic. So the baseline cycling rate is fairly low after time in culture. We hypothesize that there may be genes that can influence cardiomyocyte proliferation when knocked out. Therefore, we decided to perform a CRISPR library screen for cardiomyocyte proliferation. We started by generating a gene list using both literature and ontology. We converted these genes to the rat homologs because we're performing this screen in rat cells. And then we designed guides targeting the first exon such that the gene is robustly knocked out via frame shift mutation. We then designed the vector, inserted the sgRNA sequences, and cloned and made lentivirus. Lentivirus inserts into the genome, and it's very valuable because you can sequence using next generation sequencing at an early time point and a later time point after proliferation has occurred, and determine which sgRNA changed over time. The ones that are overrepresented over time in culture would be the ones that conferred either a proliferative advantage or survival advantage to cells. To narrow down our gene list, we first started with cell-cell and cell matrix adhesion proteins. We decided that to use these types of proteins because we hypothesized that alterations to these types of proteins may be more likely to result in cardiomyocyte proliferation. We then removed genes that aren't expressed in the human heart because it doesn't make sense to knock out genes that aren't already expressed. We then removed ion channels and non-membrane proteins to further narrow the list. This resulted in a 237 gene library, which is small enough that we could have very high coverage of this library. High coverage is especially important because we didn't expect large clonal proliferation events because the cell cycling rate of these cells at baseline is so low. So the higher coverage of the library allows us to get a better outcome at the end of the library screen. And what you can see here is the design schematic for this CRISPR knockout screen performed in NRVMs. So we started with the P2 NRVMs mentioned earlier. After 3 weeks in culture, which is when the cell cycling rate decreases, we transduced the cells with the library lentivirus. After 48 hours, which gives the cells enough time to uptake the virus and be transduced, but not enough for significant expression of the virus, we isolated the initial gDNA for our initial representation of the guide RNAs. We then waited 2 more weeks and did our final gDNA isolation. So you can amplify the guide RNAs out of the genomic DNA and send them for high-throughput sequencing. Then if you compare the representation at the 2-day time point to the 2-week time point, you can see which guides increased over time in culture. To do this screen, we used a cardiac-specific CRISPR knockout construct, which was modified from lenti-CRISPR V2 from the Feng Zhang lab. And we modified it to add this chicken troponin T promoter, which is a cardiac-specific promoter. This means that Cas9 will only be expressed in the cardiomyocytes, and the sgRNAs will only have an effect in cardiomyocytes. So after performing this screen, we have shown here the top 10 overrepresented genes as identified by this screen. And what you can see on the left is the robust rank aggregation score. And we use the pipeline, which is a CRISPR analysis pipeline called MAGIC. And the higher the RRA score, the more statistically overrepresented this gene is out of the 237 genes in the library. We then sought to validate our individual candidates in 3D RVM engineered cardiobundles. So to make these cardiobundles, we mix cells with a fibrin hydrogel. And at the time of making the gel, we also add in our cardiomyocyte specific knockout lentivirus for each of the genes listed below. We performed this experiment over 2 weeks in culture with EDU, which labels DNA synthesis or cycling cells from day 12 to 14 in culture. So it'll label cells that are cycling in that last 2 days of culture. And what we noticed is that the cardiobundles increased in size after that 2 weeks with the knockout virus. So what you can see here is the no virus control, and then each of these bundles had a single gene knockout. And what we're seeing here is that several of these candidates can significantly increase EDU incorporation at the 2-week time point. These are cross sections of those engineered cardiobundles with nuclei in blue and EDU in green. And what you can see is that several of these genes significantly increased EDU incorporation within cardiomyocytes. And our no virus and non-targeting sgRNA controls weren't different compared to each other. We also used SAB1 as a positive control for cardiomyocyte EDU incorporation because we knew that this gene should increase cardiomyocyte EDU incorporation. What we saw was that only a single gene significantly increased F-actin positive area, which should label the cardiomyocytes at the 2-week time point, and that was ADA, or adenosine deaminase knockout. And what you can see here is several of the bundles may appear larger, but there is more interstitial space in some of these, resulting in no significant increase in F-actin area despite the apparent larger diameter of the cardiobundle. We then sought to do a time course for the EDU incorporation and cardiomyocyte number with these gene candidate knockouts. So what you can see on the left is cardiomyocyte nuclear number per cardiobundle cross-section, and on the right, it's cardiomyocyte EDU positive nuclei. What we saw is that only some of the genes increased either cardiomyocyte EDU or cardiomyocyte nuclear number. However, it looked like adenosine deaminase had the most positive or promising trend, with the number of cardiomyocyte EDU positive nuclei increasing over time, despite the controls, both the no virus and non-targeting controls, decreasing over time in culture. The data shown here on the left are representative images of that same data from the previous slide, where you can see what the cardiobundles looked like in cross-section over 3 weeks in culture. So the no virus and non-targeting controls slightly increase in size over time, but modestly. However, the one that was most striking was adenosine deaminase knockout, where the cardiobundle appeared to increase in size over time, which correlates with the cardiomyocyte EDU and also the fold change in cardiomyocyte nuclei. So the number of cardiomyocytes is increasing, and the number of EDU positive cardiomyocytes is also increasing with adenosine deaminase knockout. We wondered then whether adenosine deaminase knockout may affect the ultrastructure of these NRVMs shown here in monolayer. And we didn't notice any apparent differences between the control with no virus and adenosine deaminase knockout. However, the cells were more dense in the adenosine deaminase knockout group because of the additional number of cardiomyocytes, because of the induced proliferation. All of that data shown previously was in rat cells, but of course we're interested in what happens in human cardiomyocytes. So the data shown here is in human iPSC-derived cardiomyocytes. So what we can see here, this DCF is an adenosine deaminase inhibitor drug, while ADAKO is adenosine deaminase knockout. And what we can see is that both the inhibitor drug and adenosine deaminase knockout significantly increase EDU incorporation in human cardiomyocytes. The inhibitor also increases the cardiomyocyte number, and interestingly, some doses of the drug increase cardiomyocyte mitochondria. Mitochondrial intensity is known to increase with cell cycling, but it could also increase as a result of change in metabolism. So we'll be interested to look further into this change. Another thing that's very important to consider is that cardiomyocytes can become polyploid, and some of our mitogens may induce polyploidy rather than cytokinetic division. So we checked to see whether this increase in cardiomyocyte EDU may be due to an increase in ploidy. And it appears that there's no significant difference in the percent 2N or diploid cells in cultures treated with DCF, this adenosine deaminase inhibitor. So it does appear to be inducing cytokinetic division. So in conclusion, our CRISPR screening methodology was applied successfully to identify new cardiomyocyte mitogens. And the effect that adenosine deaminase knockout had was the most prominent effect in both rat and human cardiomyocytes. In the future, we'll test if adenosine deaminase knockout can increase the size of our implanted engineered heart tissue patch grafts. And we also plan to test whether adenosine deaminase knockout in vivo via an AAV-based gene therapy is beneficial to cardiac function after MI model in mice. Thank you so much for listening to my talk today, and I'm open for questions. I'd like to thank everyone in my lab who helped with this work, especially the undergraduates.